Okay. Our next presenter is Maria Jose Contreras Zarate from the University of Colorado, presenting GS 507. Estradiol represses antitumoral immune response to promote progression of triple-negative breast cancer brain metastases.
Thank you. Good afternoon. My name is actually Diana Cittelly. On behalf of Maria Jose and the rest of my co-authors, I'd like to thank the organizers for the opportunity to present here today. I have no disclosures. So as you all might know, younger age is a significant independent predictor for the development of brain metastasis in breast cancer. And we have often thought about the risk of brain metastasis being increased in younger women as a result of the increased incidence of very aggressive triple-negative breast cancers in this population. However, the incidence of brain metastasis even within patients with triple-negative breast cancer is higher when women are diagnosed in a premenopausal stage versus postmenopausal women. So we have long investigated the hypothesis that premenopausal hormones, such as estradiol, might promote brain metastasis by exerting effects on the microenvironment. My lab has recently demonstrated, actually, the astrocytes within the brain microenvironment express estrogen receptors. And through this interaction of estradiol with those estrogen receptors, upregulate the secretion of a number of growth factors, including BDNF and EGF, which then in turn activate oncogenic signal within triple-negative breast cancers, such as EGFR and TRKB, that increase migration, invasion, proliferation, and ultimately increase brain metastasis in triple-negative breast cancers. However, we also know that the brain is somewhat unique in that it also produces estrogen, and that reactive astrocytes surrounding brain metastasis express aromatase and can synthesize estradiol, regulating very important functions within the brain. So we know that both ovarian estrogen as well as brain estrogen might be contributing to the progression of brain metastasis. A few years back, we have actually demonstrated that estradiol indeed increases the colonization of the brain in a model of triple-negative breast cancer cells. So in this case, we injected triple-negative 231BR cells in ovarectomized female mice that had been then treated with premenopausal levels of estradiol, so that we will make premenopausal hormones in females, vehicle, or the aromatase inhibitor letrozole, so that we could block any effects of peripheral estrogen. And what you can appreciate here is that premenopausal levels of estrogen cause an increased frequency of metastasis. This was measured by MRI at the time. We have the same similar results with histological quantifications. And we can see also that both ovarectomy or the combination of ovarectomy with letrozole significantly decrease the ability of those triple-negative breast cancer cells to colonize the brain, which, clinically speaking, is an indicator that estrogen is able to promote the ability of cells that have seeded to the brain to actually form metastasis. However, we know that it's unlikely that estrogen depletion therapies can be tested clinically in a prevention setting. So the goal of this study was to test whether estrogen depletion therapies alone or in combination with current standard of care could decrease progression of existing brain metastases in models of triple-negative breast cancer. To do that, we first started with a very known and very aggressive model of triple-negative breast cancer that is syngeneic to BALB/c mice, which is 41BR5 cells. You will see here that this study is slightly different to the one that we have done before, in that we overectomized the mice and supplemented them with premenopausal levels of estrogen, injected the cancer cells, and then allowed these metastases to form for 7 days. In this model, this is the time where metastases are already seeded and they are outgrowing as large metastases. At this point, we randomized the mice into either continue receiving estradiol, going into estrogen withdrawal alone, or estrogen withdrawal in combination with letrozole. And because we know that the standard of care for the majority of patients with brain metastasis includes brain radiation, we gave the mice a single dose of 15 grays, which is not lethal for this strain. And then we analyzed metastatic burden. And as you can see here, we see a suggestive decrease on the number of metastases or the extent of metastatic burden, intracranial metastatic burden in this mouse, only in the mice that were treated with estrogen withdrawal and letrozole in combination with 15 grays of radiation. And here is an example of the metastatic burden. The brightness shows some of the tumor burden in the brain of these mice. Even though this didn't reach statistical significance, we conducted a similar experiment using the E0711 model, which is syngeneic to C57 black mice. We use exactly the same experimental setup. And I want to clarify here, 14 days is the time where we have to euthanize the mice due to extensive tumor burden. So this is a rapidly aggressive tumor where 7 days are really modulated, mimicking women with symptomatic metastasis. And as you can see here, in this model, we see a more robust decrease in metastatic colonization progression, particularly intracranial progression, both with mice that were treated with estrogen withdrawal alone or estrogen withdrawal with letrozole. And surprisingly, we see also a response in terms of systemic metastasis in these mice, which we presume is the result of an immune component to this effect. Because we also know not everybody receives radiation, we conducted parallel experiments in which we remove the 1 dose of 15 brain radiation in these mice. And for our surprise, we saw absolutely no effect of estrogen withdrawal or estrogen withdrawal with letrozole, either for intracranial disease or for the systemic disease in this model, which suggested that the effectiveness of estrogen suppression to decrease progression of existing metastases depends on priming of the immune system by radiation. To test this, we decided to inject the cells that were responsive, the E071 cells, into NSG mice. And we performed exactly the same experiment with the only modification that the mice only received 10 gray radiation as larger radiation doses are lethal for these mice. And as you can see here, we don't observe any effect of estrogen withdrawal or estrogen withdrawal with letrozole, both when there is radiation on the experiment or in the absence of radiation in parallel experiments. Again, suggesting that this is an immune-related response. And we have observed similar results when we use human xenograft models of PDXs in NSG mice. So again, we concluded from this that the effectiveness of estrogen suppression to decrease progression of brain metastasis depends on a fully functional immune system. Now, we have gone on to try to define how estrogen and hormonal therapies could be influencing the brain immune response associated with metastasis in these estrogen receptor negative models. And for this, we use, again, the model of the E0711 cells into C57 black mice that had been ovexed and treated with the different hormonal responses and analyze brain immune infiltration very early after cell dissemination, in this case, 3 days after injection. And I hope you can appreciate here that one of the major findings is that estrogen represses microglia activation or microglia recruitment to this activation in the brain, and that ovarectomy alone or in combination with letrozole significantly increases the amount of microglia. This early activation of resident microglia is a hallmark of an effective brain immunosurveillance and might limit the success of early disseminated cells to colonize the brain. By contrast, as we let the animals develop larger metastases and we see a later event within that metastatic progression, we observed the already reported switch to a tumor-induced chronic inflammation, particularly in the estrogen-treated mice. That is marked here for this increase in microglia and macrophages, and a significant reduction in those mice that have been treated with ovarectomy alone or in combination with letrozole. Importantly, we observed a significant increase in the fraction of T and B cells that were recruited to this brain microenvironment in mice that had been treated with ovarectomy alone or ovarectomy with letrozole, suggesting that some of the response that we are observing in terms of decreases of metastatic progression in that setting might be due to an effective immune response. Now, we understand that there is a new approved drug as immunotherapy, and the fact that we observe recruitment of T cells, I didn't show this, but none of these T cells show significant differences in the levels of PD-1, which suggested to us that we still, even though we have an increase in T cells in that microenvironment, this increase in T cells might not be necessarily cells that will be totally functional. So we did a pilot experiment in which we combined estrogen or estrogen withdrawal with letrozole with anti-PD-1 antibody for the mouse. And what you can see here in this experiment were initially not significant difference between the treatments. I want to note that this model progressed really fast, and we lost 4 animals in the estrogen group, presumably for increased metastatic burden. The fraction of a stable disease in the brain, though, was similar between the estrogen-depleted mice and the estrogen PD-1, estrogen withdrawal plus PD-1 treated mice. So in this fraction here, you will find the animals that, or the initial metastatic burden that these animals have, and we see similar fraction between these PD-1 treated with estrogen or the estrogen withdrawn with letrozole. And we see similar results for systemic metastasis. So at this point, we don't see additive results between effects between combination of PD-1 and estrogen depletion in this study. So as a conclusion, I hope I provided enough information to show you that premenopausal estrogen levels promote an immunosuppressive brain tissue microenvironment that contributes to brain metastasis progression, and that estrogen depletion therapies reactivate an antitumoral immune response, including increased recruitment of T and B cells, that estrogen depletion might be used in combination with SRS to decrease brain metastatic progression in younger women with triple-negative breast cancer with and without extracranial disease. But further studies are needed to determine the benefit of estrogen depletion therapies in combination with immunotherapies. I want to highlight the need for basic researchers to use a combination of preclinical models mimicking standard of care in brain metastasis research. And with that, I want to thank my lab, all my collaborators, patient advocates, and funding sources. Thank you.
Thank you very much. While people are moving towards the microphones, I will take one question online. For the clinicians in the audience, how do you localize an entire gene expression profile to individual cells on an H&E slide?
That might not be a question for me.
I think that's a prior.
That was for the previous.
Oh, that was a prior. Oh, no. Sorry, the screen has not moved around. Okay, we have question microphone 3, please.
Just 2 questions. First one is, do you have any idea which receptor is responsible for the estrogen response you have observed? And the second question is, have you tried CD8 depletion to see if the effect is dependent on? I mean, you did do compared with the regular xenograft and the immune proficient model. So just those 2 questions.
So first question, we don't know yet exactly what receptor is. There is estrogen receptor alpha and beta also in astrocytes. We have current studies in which we can selectively knock down estrogen receptor alpha in the astrocytes, and we are trying to run those studies. I can tell you ahead of time that not all of the effect is mediated by estrogen receptor alpha, and that we can see lack of sensitivity to estrogen, but not necessarily the same influence in metastasis. We don't know yet whether estrogen receptor beta has any effect on that. We are conducting those studies. And the second question was about CD8 cells. Sorry, can you repeat me the second part?
CD8 depletion by using CD8 antibodies?
Yes, we haven't done that, but that's one of the things that we have planned. We wanted to initially test the PD-1 just for the translational potential, because we know that PD-1 has been recently approved for triple-negative breast cancer, and we have to, this is a preliminary experiment that we also have to repeat.
Thank you. Excellent.
Okay.
Thank you.
I'm going to take number 3 again, because I know you were waiting last time as well.
Yeah, thank you very much. Olopade, Chicago, really very nice talk. 2 questions. One has to do with whether you plan to do either single-cell transcriptomic or spatial transcriptomic to know the crosstalk between the microglia cells and the response. And then secondly, is the response subtype dependent?
Well, the first question is, yes, we are doing right now, we are doing a single-cell RNA sequencing on isolated brain cells, because as you can appreciate, there is a lot of heterogeneity and phenotypes within brain immune cells. We are learning a lot about also brain-specific responses, so we hope to be able to be more specific about the populations of cells associated to this phenotype, at least in the mouse. We have plans to do similar experiments with human samples using other methods. And we think that the implications of the studies are that, one, we could use, in theory, estrogen receptor inhibitors, like selective estrogen receptor modulators or aromatase inhibitors, for decreasing brain metastatic progression in patients other than estrogen receptor positive breast cancer, like triple negative breast cancer. So I think that will be the major implication for this study, but speculating that this also has an effect on patients who are treated with estrogen receptor positive breast cancer.
Thank you very much again. Unfortunately, we have to move on now.